Rattlesnake. For most people, the first term associated with rattlesnake is venom. Was venom featured on a TV special that you saw? Or maybe you know someone or someone's pet that was unfortunate enough to get bitten. Indeed, their venom represents a crucial aspect of their biology, and several studies even report rapid evolution of venom genes driven by prey preference. However, the complete biological context in which venom is deployed is far more complex, and, in my opinion, fascinating. Long before a rattlesnake can even use its venom and enjoy the resulting meal, it must first detect, track down, and ultimately sense the presence of a potential prey item. Prey-based selective pressures should drive the evolution of genes responsible both for prey detection and acquisition, resulting in signals of adaptive evolution in the gene sequences underlying these traits. I personally like to think of the phrase, if you can't find it, you can't kill it, you can't eat it. Rattlesnakes sense the world through color vision, bifocal heat sensing, and dual system smell or chemoperception. The genetic underpinnings are well characterized for pit viper vision and heat sensing. However, a similar characterization of their chemosensory abilities remains undescribed. My PhD research attempts to fill this gap using some of the state-of-the-art genetic approaches that are available. And today I'm going to highlight some of my work characterizing the complete chemosensory repertoire of our Florida native rattlesnake, the eastern diamondback. I identified hundreds of genes coding for 2 different types of receptors spread across the genome. When expressed, these membrane-bound receptors detect chemical signals from their environment, shown here as red dots. I compared the gene sequences and found more variability in the parts of the receptors that interact with the chemical cues, shown here in green. This suggests that certain smells detected by the rattlesnake are likely driving the evolution of these receptors. So why does this even matter? First and foremost is conservation. By considering the mechanisms underlying how predators find their food, we can more effectively preserve the species. In addition, characterizing any biological system down to the underlying genetics enables profound crossover into other disciplines, such as medicine and neurology. Thank you for listening.